Hello friends. So we are starting with the physiology of central nervous system, and today we'll have a look into the sensory system, especially the introduction to the sensory system, and we'll look to touch upon certain areas of the somatic sensory system. Like we'll try to have an overview of the somatic sensory system, and before going to actually having a look at the somatic sensory system, we will first go through what happens in a sensory system overall. I mean, be it the somatic sensory system or be it the special sensory system. So basically, this is how the sensory system works. We all know that the nervous system can be functionally divided into an efferent system, that is the motor system, and the afferent system, which is nothing but the sensory system. The sensory system is then divided into the somatic sensory system, and the other one is the special sensory system in toto. So the somatic sensory system actually doesn't end there. So somatic sensory system is nothing but many of the sensations which is produced from the periphery of the body, or I would rather say that sensations produced from the skin and the sensations produced from the periphery of the body, such as the joints, spaces, your muscles, tendons, the stretch which they undergo. So they all come under the umbrella of somatic sensory system. On the other hand, the special sensory system, as you all know, is covering your vision, your uh, uh, hearing, the gustatory sensation, which is nothing but your taste, the olfaction, the sense of olfaction, which is nothing but your smell, and so on. So today, before going into the somatic sensory system, we'll have a look how the sensory system is organized, right from the receptors to the cerebral cortex and further. So basically, every sensory system, be it the somatic sensory system or the special sensory system, starts with the receptors. So the receptors are of different types in different sensory systems, which will be dealt later. So the basic is it starts from the receptors. So it starts from the periphery and it goes to the center, which is nothing but your afferent pathway. So it starts with the receptors, and from the receptors it is carried to the CNS. So which part of the CNS the senses enter, that will differ between the sensory or the somatic sensory system and the special sensory system. In case of somatic sensory system, it is the spinal cord or the brainstem for impulses carried by your trigeminal nerve, which will carry sensations from your face and so on. So basically it works this way. And for the special senses, say your auditory pathway for example, ends up in the auditory nucleus, which is located, or the cochlear nucleus, which is located in the medulla of the brainstem. And also the balance, the sensation of balance, which is sensed by your vestibular system, is carried by your vestibular part of vestibular cochlear nerve, or the eighth nerve, into the brainstem. That is the vestibular nuclei of brainstem, which are organized again into the four different groups of nucleus, which will be dealt later. And but few special sensations doesn't obey this rule. For example, your olfactory sensations doesn't have a brainstem or spinal cord center. Again, your visual sensation doesn't have a brainstem or spinal cord center. They are integrated or organized in a different fashion. So what are the other special sensations left is nothing but a gustatory sensation, which is carried, as we all know, by the chorda tympani branch of facial nerve from the anterior two-thirds. The posterior two-thirds is taken by the glossopharyngeal nerve, and the posterior most part of the tongue. The taste sensations from the posterior most part of the tongue is carried by your vagus nerve, and they are all integrated in nothing but the nucleus tractus solitarius, which is present in the middle of the brainstem. So basically, the first order neuron in any sensory pathway comes from the spinal cord of the brainstem. So that's what I'd like to tell. But they have few exceptions as well, so it differs from one place to the other. And the first order neuron ends in close contact with the receptor, or in case of the somatic sensory system, the nerve endings of the primary neuron or the first order neuron are themselves modified into the receptor, as we'll say later. So okay, the sensations are taken care of by the first order neuron and they enter the central nervous system. So where they go next? So they, from the first order neuron, the first order neuron gives rise to the axons, as you know. So they will synapse with the second order neuron. The axons form tracks, and these tracks ends in thalamus. So which nucleus of thalamus? Again, it differs between sensations. For the somatic sensation, it is the ventral posterior medial nucleus. For the auditory sensation, it is the medial geniculate body, and so on. So basically, the first order neuron synapses with the second order neuron, which is present in the nuclei of thalamus. Okay, so it has ended up in thalamus. So where will the sensations go next? It will go into the cerebral cortex, and the cerebral cortex gives rise to the third order neuron. So I hope it's clear the basic framework of the sensations, which is nothing but how the sensations are carried from the periphery to the central nervous system. So in the cortex there are different areas which will deal with different sensations, as you all know. So the basic idea is the receptors, and from there, from the receptors, it enters the spinal cord, which gives rise to the first order neuron. And receptors, from the spinal cord, it goes to the thalamus, which gives rise to the second order neuron. From the thalamus, it goes to the cortex, which gives rise to the third order neuron. And this is a basic framework which will have a slight modification here and there for different sensations. So okay, I think with that it is clear that the afferent pathway starts with the peripheral receptors. Okay, so as I told you before, the senses are broadly classified into somatic sensations and the special sensory sensations. Okay, what comes under the somatic sensations will be the next question in our minds. And under the somatic sensations, it is nothing but your touch, pressure, pain, and temperature. And all of these, as you all know, are sensations that are arising from the skin. And there are actually a special subgroup of somatic sensations which are also called as the synthetic or cortical sensations. Okay, what does these mean? These are actually a very high level kind of the somatic sensation. Like the earlier set sensations like pain, temperature, touch, or I would say the crude touch, have existed for a very very long time in the history of evolution. But there are certain sensations such as your fine touch, and recently, as the human beings continued to evolve, they started acquiring the uh, the apparatus to sense newer sensations. So what are these sensations? These sensations are nothing but synthetic or cortical sensations. That is, they need the cortex, the integration of the cortex to pick up these kind of sensations, and the further analysis has to be taken place in the cortex so as to provide some sort of meaning for the sensations to be carried. So what are the sensations? These are nothing but your vibratory sensation, and these are your stereognosis, which is nothing but you ask a person to close his eye and give him an object, ask him to just identify the object by touching, which obviously involves a bit of the memory and also the ability to touch the object. And this touch, or the higher level of touch, which is recently evolved, feature or a sensation is coming under the cortical sensation. And the other is 2-point discrimination, which is also a very localizing feature of the which involves the cortical function, because without the cortical function you can't localize, and when you can't localize you can't discriminate sensations arising from between the two closer points. So I hope this is pretty clear for all of you. And with that I think we can go on to the organization of the somatic sensory system, which is nothing but which starts with your receptors toto. Okay, what are the types of receptors? So okay, this receptor types here given here I have described in reference to both the somatic sensory system as well as the special sensory system. For example, the chemoreceptors are concerned in both the somatic sensory system as well as the special sensory system. In case of somatic sensory system, I would rather say chemoreceptors are not that active, but they modulate the responses to the impulse transmission, such as the pain sensation, where the pain is actually the endings are actually more sensitized to pain on exposure to certain chemicals such as your bradykinins, the prostaglandins, so on. And most importantly, the chemoreceptors are involved in olfactory pathways and also the gustatory pathways. So coming next is your pain receptors, which respond to tissue damage. So what is a function of pain? Anyone would ask me, I would rather say it is an indicator of tissue damage, so that the person is aware that some kind of damage is going on. Just get your body away from that sort of damage. So it is a warning signal. Pain is a warning signal. And up next is here thermoreceptors, which obviously, as the name says, thermos temperature, and the receptors which senses the temperature changes. Like we have the warm receptors and also the cold receptors. And next is your mechanoreceptors. So the mechanoreceptors actually take part in feeling changes at the surface of the skin, like any object moving on, or it can kind of identify the vibratory sensations, or it can even find the borders of an object. It can differentiate between objects. So how come a single mechanoreceptor type category can, like, it is a very simple new structure, but how does it do all this special function? We'll deal with it later, because the mechanical receptor endings or any receptor in the central nervous system have features called adaptation. So under the mechanoreceptors, as we'll see later, there are different kinds of receptors with different adaptation times and different anatomical structures as well, which enable them to carry out the different functions. Up next is your photoreceptors. As you would see, the rods and cones of the eyes are nothing but your photoreceptors. And osmoreceptors, okay, well, where is the osmoreceptor located? As you all know, then our osmolarity of the blood increases. There are certain areas in our brainstem as well as in the brain which are not guarded by the blood-brain barrier. So we have the osmoreceptors, which senses the changes in osmotic pressure, or the osmolarity, I would say, and send signals to the hypothalamus to reduce antidiuretic hormone from the posterior pituitary. So as I told you before, mechanical receptors, though the category looks simple, it the kind of sophistication that leads them to carry out different function is enormous. Like for example, you can see here that the Meissner's corpuscle, Vaccinian corpuscle, and Ruffini endings have something like a capsule here, whereas the Merkel cells lack the capsule. So In general, mechanical receptors can be classified broadly into two categories, which are nothing but your capsulated ones, like the Meissner's corpuscle, Paxinian corpuscle, Ruffini endings, and the non-capsulated one, like the Merkel cells. Okay, what are their functions? There you go. First, coming to the Paxinian corpuscles, these are located in the deeper subcutaneous tissues, tendons, and ligaments. They detect heavy pressure and vibrations. As you will see later, Paxinian corpuscle has a very short adaptation time as compared to other receptors. So this short adaptation time is important for picking up the vibration sensations. Okay, what do I mean by adaptation? Uh, I would rather explain it with a simple analogy. You wear a shirt for the first time, your skin picks up the sensation and it will feel weird for some time. But after some time, you don't realize that you are wearing a shirt or anything for that matter. So this is nothing but adaptation. Basically, you the nerve endings at first fire rapidly as soon as they get any kind of stimulus, but once the stimulus is keep on stimulating the nerve ending continuously, they go for something called an adaptation, which is not fatigue exactly, but we can call it that way as well. So they stop firing, they are not excited as they were before, so they like kind of produce a response which is submaximal. And that's why I would say adaptation is a special feature for vibration. Why? So basically, if the nerves are very fast adapting in nature, you have to give a stimulus which has breaks obviously, but in it should be spaced very shortly, that the time limit between one stimulus to another is very short. So this kind of looks like a vibratory stimulation, right? Why can't nerve ending with a very long adaptation time pick up this one? Let's consider Meissner corpuscle, for example. So basically, the Meissner corpuscle is helpful in detecting the fine touch and distinguishing between the 2 points on the skin, and they adapt rapidly. So even here, it can pick up vibrations, but not to the extent of the Paxinian corpuscle. It can pick up vibrations, but at a smaller frequency. I would go to the free nerve ending or expanded tip receptors, which take a very long time to adapt. So what happens as a result of very long time of adaptation? So basically, if it takes a very long time to adapt, you have to space your senses or the stimulus in such a way that the interval between one stimulus to other is greater than that of the adaptation time. And vibration senses, as you all know, the time between one stimuli to the other is very short. So if this is the case, how can the nerve endings with very long adaptation time pick up vibration? So that's why the nerve endings which adapt very rapidly are apt for picking up the vibrations. So the Paxinian corpuscle is ideal for picking up the sensation of vibration. And as you would see, Paxinian corpuscle is located in the deeper subcutaneous tissues, tendons, and ligaments, whereas the Meissner corpuscle is present on this body surface, and it will adapt rapidly, yes, but not to the extent of Paxinian corpuscle. So basically, the Meissner corpuscle detect fine touch. It will distinguish between 2 points of the skin. All right, it can do all that, but it can't. It is not that effective in picking up the vibratory sense. And then we'll move on to the free nerve endings. They say these free nerve endings are mostly the warm, the warm receptors, cold receptors, and they pick up the primitive sensations such as itch, tickle, and even some say they pick up the pain sensations as well. Up next is extended tip receptors or ecgodome receptors. The name because why it is called ectoderm receptors? The Merkel disc or the expanded Merkel discs or resembling the ectoderm receptor. I can't actually show this. You have a very good picture in Guyton's textbook of physiology of the ectoderm receptor, where they show all your beautiful explanation why it is called the ectoderm receptor and why it is not apt for adaptation, or why it is not apt for picking up the faster stimuli. It is the reason they adapt initially very strong, but after that they don't completely adapt. So this is the reason. And also they are helpful in picking up the touch and texture. So texture of object is mainly picked up by the Merkel's corpuscle. Like for example, what is texture? You have a silk object, it will feel different to you. And you have an object made of glass. How the surface of glass appears to you? It is very quite smooth and so on. And you take soil particle, it feels rougher to your hands, right? So this is nothing but your texture, and it is picked up by your Merkel's corpuscle. So what actually is picking up the objects or stimulus which is running over your body? Like for example, let us assume that there is an object running over body like an insect crawling over us. So it is picked up by the hair end organ, which adapts rapidly, which adapt rapidly and basically help in picking up objects which is running rapidly over your body, as well as the initial contact it makes with your body. What then is the help of Ruffini ending? The Ruffini ending, again, they say they are not much involved in the surface sensations like your pain, touch, temperature, itch, and so on. They are much involved in a deeper sensation like the proprioception. I think with that we can actually move on to the next topic, or before moving on to the next topic, you'll have a quite a small overview or a look into how the mechanical impulses are transcribed into nerve impulses. So see here, we have a Paxinian corpuscle end organ, and as you would all know by now, it is a capsulated end organ. So basically, and deformation on the capsule leads to stretch of certain receptors. Basically, it is not the receptor, certain ion channels. So as you would all know, there are three broad categories of ion channels, which are nothing but the mechanically gated ion channels, and the chemically gated ion channels, and also the voltage gated ion channels. So the one responsible here is a mechanically gated ion channel. So what happens is, as a deformation to the capsule of Paxinian corpuscle occurs, the deformation is transmitted to the mechanically gated ion channel in the nerve ending, and as a result, the mechanically gated ion channel opens and lets sodium inside. So what happens as soon as the sodium enters the cell? It depolarizes. So usually the resting membrane potential is somewhere around minus 70 microvolts, and as soon as the sodium enters, it goes to the positive direction. So this is called as depolarization, as you all know. But will this be enough for causing an action potential and producing a nerve impulse? Absolutely not. So for that, there are two basic concepts. It's nothing but the temporal summation and as well as your spatial summation. So what is temporal summation? Basically, at so basically, let us assume the x-axis is time and y-axis is voltage. Okay, so the resting membrane, this is 0, and the resting membrane potential is somewhere around here. Let us consider, for example, okay, I'll draw in a different color. So resting membrane potential is somewhere here. As soon as the excitatory potential or the threshold happens, what happens is it moves a little towards the positive direction and it comes back. Okay, so if we give stimuli from different direction to the corpuscle, so spatial summation is basically this: we give different stimuli at the same time at different spaces on the capsule. So what happens is, this is response for one stimulus. If I summate down to the same thing, so basically it grows in size, and it will, at some point it will reach the threshold. Let's assume the threshold is somewhere here. Let me call it thresh. So as soon as it reaches the threshold, what happens is there will be an upstroke, and it will go beyond, it will reach the peak, and as it repolarizes, it will go below the resting membrane potential, and as a result of the action of sodium potassium ATPase pump, it will come back to its original location. So this is your spatial summation. Basically, one action potential or the one stimulus produces a local potential which is not enough for propagating the action potential. It is called the excitatory postsynaptic potential. In a nerve ending, here it is an excitatory action. It is an excitatory local potential, not an action potential. So as a result of some spatial summation, more the potential, more EPSP act at the same time, and as a result of which there will, the voltage or the membrane voltage goes closer to the threshold as a result of spatial summation, and once it reaches the threshold, it does upstroke and comes back to the RMP, and this upstroke is enough for propagating the action potential, and as a result of which a nerve impulse is generated. Okay, what next is temporal summation? I would like to do that. Temporal summation with the basic concept. Okay, there is an upstroke here. Okay, then I give another stimulus before this dies down. This will usually die down. Okay, assume this dies down, but I give, before it dies down, I give another stimulus. It will die down subsequently, and before it dies down, I give another stimulus. So slowly and slowly, as I give and stimulus before the initial or the earlier excitatory potential dies down, what I am effectively doing is summating all the excitatory potential, and once they summate above and threshold voltage, what happens is basically the excitatory peak, then a fall, then the action of sodium potassium ATPase pump, and it will come back to normalcy. So this is nothing but a temporal summation. So I hope the concept of the excitatory potential, the spatial summation, and the temporal summation are pretty clear right now. With that, I think our receptor concept is over. So basically, the sensory pathway starts with the receptor, and in case of somatic sensory system, we have basically two kinds of the main kind of receptor is nothing but your mechanoreceptors, the thermoreceptors, and your nociceptors, which are the pain receptors. And in case of mechanoreceptors, they are again classified based on the structure into two: capsulated and encapsulated, and all the mechanoreceptors have different adaptation time. These kind of differences in adaptation time and the structure gives them different functions, like the Meckel's corpuscle, for example. Meckel disc, I would rather say, for example, is helpful in picking up the touch, localizing the touch, and determining the texture. The Meissner's corpuscle is useful in detecting fine touch and for 2-point discrimination. And the air end organ, for example, is helpful in picking up the touch, but more importantly to pick up objects which run over the surface of your body. The Paxinian corpuscle for vibrations, the Ruffini endings for deeper sensations such as the proprioception. So with that, I think the receptor concepts are clear, and